So for today's assignment, what we're going to do is we're going to access the virtual lab gel electrophoresis. And what a gel electrophoresis is a test to determine how we analyze DNA found at a crime scene. So what you're going to do is you're going to go to our Schoology and you're going to look for the virtual lab electrophoresis document file. You're going to click the link that's underneath the title, and it's going to take you to this website. Now, if it prompts you to access Adobe Flash Player, go ahead and click that and allow. But what we're looking for is gel electrophoresis, which suspect's DNA matches the DNA found at the crime scene.
A local neighbor.
Now, what you're going to notice is that this particular software will talk to you, or you can just simply pause it and just read it through yourself. So you're more than welcome to do that. But the important thing is we got to figure out what the problem is. So hair was found at a crime scene where a bicycle chain was broken, and it looks like the bike has been stolen. So we want to make sure that we're able to tie an individual to this particular crime.
Explore the lab to learn what.
So all you're doing at this point in time is going through and making sure that we know what type of items that we're dealing with in terms of this virtual lab. And what the interesting is about this checklist is that if you highlight over each one of these items, it'll tell you what they are and where they're located. So if we're looking at deionized water, we're going to go back to our document file, and we want to make sure that we know what each one of these different items are. So you don't have to know every single item, but let's say, for instance, agarose gel. So we want to make sure that we click the agarose gel. And in terms of the agarose gel, we want to write down this information associated with agarose gel. So you don't have to necessarily write down a piece of paper. You can either copy and paste it if it allows you to copy and paste, or you can just simply type it out. Okay, but you are going to be turning in this electronic version for this particular assignment. So we're going to go ahead and just click through each one of these things.
Gels, deionized water, ultraviolet, TB, ethidium, bromo, micro, digest, pipette tips, power supply, electrophoresis chamber, digested DNA sample, DNA markers.
Okay, so those were all the different items, and you're going to have to click procedure after that.
You will produce it.
And once you click the procedure, here's where we start to figure out what we're doing and what the procedure is in order to analyze DNA. So we're going to click this lab notebook. Now, you are not going to be graded on this lab notebook, so it actually doesn't really matter what you write in here. But if you wanted to answer the question, explain how comparing DNA fingerprints can help identify a person who has committed a crime, you could just simply write DNA is an individual piece of evidence that ties only one individual to the crime. So what you're going to notice once you type something, that this arrow is going to be glowing now. Until you fill out that lab notebook, you won't be able to move on. So you want to make sure to fill out the lab notebook each time it prompts you to do that.
The agarose gel is in the electrophoresis chamber.
So at this point in time, we're just going to go through the lab. So make sure that you follow the document file. So explain how comparing DNA fingerprints can help identify a person who has committed a crime. So that was the first question that we saw there. So whatever you write for that particular question is what you're going to write in here instead. So the lab notebook, again, is just to advance the actual procedure itself. But at the same time, this is what you're going to be graded on. So you're going to go through the steps. You're going to write down kind of what each one of the steps is, what each one of the steps are. I don't want you to write down word for word. Just kind of give me an idea of what is happening. Take a screenshot and paste the images below in terms of the results. So once you actually see the results, once they have the comparison of all the DNA samples that are being run, then you want to make sure to take a picture of that. Purpose of the markers, direction that DNA travels, how does agarose gel separate DNA as it travels down the electrophoresis chamber. All those things are going to be important in making sure to analyze. So again, if you have any questions, if you're not quite sure what to do for this particular lab, come find me at Microsoft Teams. Either send me a chat, or you can also talk with me through video. All right, guys, thanks.